This lecture continues our introduction into the four macromolecules that make up life. Specifically, this lecture is going to focus on proteins and nucleic acids. With a protein, proteins are made up of carbon, hydrogen, oxygen, and nitrogen. The primary function of proteins in the cell is to make chemical reactions happen. Proteins have unique three-dimensional shapes that enable them to carry out chemical activities in the cell. They're also what makes up the majority of structures found in living things. For example, this is a picture of a transport protein. It is a channel made of protein that enables things to move in and out of a cell. Proteins are also used in cell communication. Cells are going to talk to one another by transporting proteins between one another. And anywhere there's locomotion, such as a muscle cell, a heart cell, or a flagella or a cilia on a bacteria, proteins are what are making up those structures and enabling them to move. So let's look at the specific structure of a protein. The monomer of a protein is known as an amino acid. A reminder that the monomer is the one singular small unit, and we can combine multiple monomers together to make a polymer. It is called an amino acid because it has an amino region, an NH2 group. It can also become an NH3. And it has an acidic region. It has a carboxyl group, a C double bonded to an O with a hydroxyl. Since there's an amino, and an acidic group, acid, we call it an amino acid. Those groups are attached to different sides of a central carbon, and below that central carbon is an R, also referred to as a side chain. An R is like an X in algebra. It's an insert other molecule here placeholder. For example, you can have an R group like this, where there's a CH3 attached at the bottom. Or like this with just a hydrogen. Or like this with multiple CH3s. Or with a hydroxyl and a CH3. There are 20 different amino acids, and it is that unique R side chain that makes them unique. Now, with macromolecules, we always want to think, how are those monomers going to bond together into a polymer? With these, it's going to be just like any other dehydration reaction. The hydroxyl at one end and the hydrogen at another are going to be pulled out to form a water to bond the two molecules together, resulting in this, something we call a dipeptide. In the case of proteins, when we bond amino acids together, we refer to that unique bond as a peptide bond. We call this a dipeptide because there are two molecules bound. Now, when dealing with something like this, where you have multiple amino acids stuck together, it is important for you to be able to identify the components of each of the two amino acids stuck together. Here's what I mean. For this dipeptide, let's first identify the amino group. I can see an amino group here on the left. Where's the amino group for the other amino? It is right here. Now, it is only an NH because we lost that other H when we did the dehydration reaction. Same thing for the carboxyl. You can see the full carboxyl group on this end. Here we only have the C double bond O because the OH was removed when water was formed. And of course, there are your unique R chains. So if I were to ask you where did this bond occur, it would occur right there between a carboxyl and an amino. These dipeptides form together in the very long chains called polypeptide chains. And this is just one small level of the multiple levels of organization proteins go through. Just another reminder that it is dehydration synthesis that is causing those peptides to bond together. The carboxyl group at one end and the amino group at the other end are going to bond. The hydrogen from the amino is removed and the hydroxyl from the carboxyl group is removed to form a peptide bond. Something unique with proteins is they will form sulfur bonds. They will use sulfur hydrols as H groups, and that is a unique identifier for proteins. Other macromolecules do not use sulfur. Now, in biology, the structure determines the function. And with proteins, this is most especially true. Proteins have unique three-dimensional shapes that are going to determine what they do. For example, on the left, you can see hemoglobin. This is a protein found in red blood cells. Its unique 3D shape enables it to capture oxygen and move it around your body. In the middle, this is pepsin. Pepsin is a protein found in stomach acid that helps break down protein. If pepsin sounds familiar to you, it is because it is one of the original ingredients of Pepsi. Soda was invented not as a tasty drink, but as a way to make medicine taste good. That's why even today we still have soda fountain candy shops / drugstores. You may have seen a soda fountain drugstore before. I know there's one in Orange. Pepsi was formed as a way to help you with indigestion. Now, of course, Pepsi doesn't have pepsin anymore, but they stuck with the name Pepsi. And then on the right, you can see collagen. This is a three-dimensional shape that gives structure and support to different tissues around our skin. Now, a protein isn't only a polypeptide. I showed you that peptide bonds can form together to make a long chain. This is a level of organization we refer to as a protein's primary structure. A protein can actually fold 3 more times to make more complex 3D shapes like I just showed you with hemoglobin and pepsin. If I were to fold this primary structure, I could form a protein's secondary structure. You can see it can be pleated, kind of like a folded sheet of paper, or it can be formed into a helix. The way this bonding occurs is with hydrogen bonding. Hydrogen bonds will form between an amino group and a carboxyl group, enabling this folding. Now, proteins, they're complex in 3D. They can fold yet again into a tertiary structure. The tertiary structure gives it its unique 3D shape. This is formed by R groups bonding with each other, and those R groups being unique with each amino acid can cause very interesting interactions. Sometimes they'll bond together. Other times they can actually repulse one another to make a different three-dimensional shape. If multiple tertiary groups fold together, we end up with a quaternary structure. The quaternary structure is just multiple tertiaries folded together. Why all these folds on folds on folds on folds? Well, chemical reactions. If I'm going to do a chemical reaction, meaning I'm going to build something or break something in the cell, I need a unique three-dimensional object to do that, something that will capture what I'm going to break or what I'm going to build. This is also for cell transport. If I'm going to move something through the cell membrane, I need a unique 3D tube to move things in and out. Now, something to keep in mind is College Board loves to ask questions about what if this goes wrong or what if that goes wrong. What if some of these bonds were to break? Well, if a protein loses its shape, it loses its function. Structure determines function. So if I were to have a breakage in the hydrogen bonds at the secondary level, now I only have a primary protein. If the quaternary structure were to fall apart, now I'm left with a tertiary. It is good to know what kind of bonding happens at each level, because College Board loves to ask questions about what if secondary is damaged, or tertiary or quaternary, what would be the effect? Proteins are, just like living things, are very, very sensitive to the environment. For every protein, there is an ideal pH, temperature, salinity, and concentration that it can work its best at. You can see this in this graph up here. Enzyme A works really well at a pH of around 6 point something, whereas the red line here for enzyme B works really well a little over 10. But notice what happens if it gets too acidic or too basic. The enzyme stops working. That is because it's denaturing. It is falling apart. So it is significant and important for you to know the environmental conditions for a protein, what is the most optimal for it, because once it's denatured, it no longer works. This is why if you have a fever that runs too hot for too long, it is detrimental to your health. The proteins in your body are denaturing and no longer able to function. So let's now look at nucleic acids. Nucleic acids are the genetic material for living systems. They store information and they're able to pass it down from generation to generation. Nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. If you see phosphorus, that's your ticket to know that you are dealing with a nucleic acid. As I mentioned before, this is the molecule of information. Best to think of nucleic acids as a book. It's something that's going to transport information again and again. It is found in the nucleus of eukaryotic cells and free-floating in prokaryotes. So let's look at the monomer of a nucleic acid known as a nucleotide. A nucleotide has 3 parts. On the side, you will see a phosphate group. A phosphate group gives it structure, and notice that the phosphate has a charge. It is slightly negative. That is significant for when we get to biotechnology. Attached to that phosphate group is a sugar, and the type of sugar that's attached determines what kind of nucleic acid you're working with. If it's a deoxyribose, you're working with DNA. If it's a ribose, you're working with RNA. Attached to that sugar is a nitrogenous base. It is a ring made of nitrogen. It is this nitrogenous base that carries information in this molecule. How does it do it? Well, in the case of DNA at least, there are 4 different nitrogenous bases. There's one called adenine, I'm going to abbreviate it A. One called thymine, I'm going to abbreviate it T. One called guanine, I'm going to abbreviate G. And one called cytosine, I'm going to abbreviate C. What's significant about this is these 4 letters each convey different information, and it is the combination of these 4 that get us all of the proteins we see in living systems. Think of it like English. English has 26 letters that can be combined in different ways to form words. Nucleotides have 4 letters in the case of DNA and can be combined in different ways to spell different proteins. Here's how a couple nucleotides could be bonded together. Here I have a phosphate, a sugar, and a base. Another phosphate, a sugar, and a base. A bond is going to form between the phosphate and the sugar. That's how these two will bond together. And this will continue to stack up. Why this stacking? Well, this is how information can be conveyed. If I read this from bottom to top, this says AGT. That's now a code that can be transmitted. Now, nucleotides, not only do they have different names, they also have different shapes. And if you have a different shape, you have a different function. There are 2 main categories of nucleotides, and it's a difference in the rings that make them up. If you look at this diagram here, you'll see that purines are made up of 2 rings. It's a double nitrogen base. Adenine and guanine are both purines. Pyrimidines, on the other hand, are only made up of 1 ring. Our pyrimidines in the case of DNA are cytosine and thymine. In the case of RNA, RNA does not have thymine. It has uracil instead. The way I remember this is the shorter word has the larger molecule. Purines, shorter than pyrimidines, but they have 2 rings. Purines have 2, pyrimidines have 1. There is a reason for this that we'll get to in a little bit. With the backbone of a nucleic acid, it is by the formation of a bond between the sugar and the phosphate. This particular kind of bond is called a phosphodiester bond, and it's what makes up the backbone or the rungs on a strand of nucleic acid. Let's build a strand of DNA. So here's one nucleotide. I have a guanine. You can see the phosphate, the sugar, and the base. I'm going to form phosphodiester bonds to make a phosphate sugar backbone, and now I have one strand of DNA. As you can see in the picture, DNA is a double helix. To build the other strand, the bases are going to complement each other. There are specific base rules. G's bond with C's, and they'll form hydrogen bonds when they do that. T's always bond with A's, and they're also forming hydrogen bonds. It is hydrogen bonding that attaches the nitrogenous bases together. Why hydrogen bonds? These are weak bonds. DNA is going to have to be opened to be read to make a protein or for DNA replication. By having a weak bond, the molecule can easily be opened and reclosed. Notice that the bond between an A and a T is only 2 hydrogen bonds, but the bond between a C and a G is 3. That will be significant later in the class. Also notice that purines, the 2 rings, only bond with pyrimidines. Why? Because this makes sure that the distance with these bases is always 3 rings. 3 rings, 3 rings, 3 rings, 3 rings. That makes sure that on both strands we always have the same width. If we had 2 pyrimidines bonded together, 2 1 rings, it would be too small, causing DNA to snap. And if we had 2 purines, 2 double rings, the rung would be too long, causing DNA to break. Another thing I want you to notice is look at how these strands, one end is labeled 3', that's what that apostrophe means, and the other end is labeled 5'. The way this is determined is by counting the carbons on the sugar. So looking at the 3' end, here I have my first carbon, second carbon, third carbon. That's why this is referred to as the 3' end. Here, counting carbons again, going clockwise, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. Another way to remember this is if a phosphate's pointing up, that is the 5' end. If the sugar is pointing, that is the 3' end. Notice that our second strand has the opposite. This 3' end here is attached to a 5' end. Same story up here. This 5' end is attached to a 3' end. This is what we mean by DNA being anti-parallel. The 2 strands carry the same information, but one is upside down. They both run parallel, but because one is upside down from the other, meaning this one starts with 5 and goes to 3, this one starts with 3 and goes to 5, we refer to it as anti-parallel. Always good to keep in mind what the directionality is and to remember that 5' goes to 3'. So let's look at our two big nucleic acids. DNA is the most popular and the one you've likely heard about. DNA stands for deoxyribonucleic acid, and it has the 4 nitrogenous bases of cytosine, guanine, adenine, and thymine. This is found in eukaryotic cells, things with a nucleus, and also in prokaryotic cells, things without a nucleus. The reason we call it deoxyribose is because it has a deoxyribose sugar as its nucleotide sugar. For RNA, RNA is single-stranded and is made up of the bases cytosine, guanine, adenine, and uracil. There is no thymine in RNA. It is only in DNA. RNA has uracil instead. RNA is used as a way of making copies of DNA through a process called protein synthesis. It is also found in a lot of viruses. A lot of viruses have RNA instead of DNA. We refer to those as being retroviruses. And we call it RNA because instead of having a deoxyribose sugar, it has a ribose sugar. So let's practice pulling together all 4 macromolecules we've learned. Looking at these macromolecules here that I've numbered 1, 2, 3, 4, 5, which of these molecules are used for energy by living organisms? Well, hopefully you picked 2. That is a carbohydrate. That is a disaccharide. The primary function of carbohydrates is short-term immediate energy. Let's try another one. Oh, sorry, lipids can also be used for energy, but that is long-term energy storage. So if you pick 2 or 4, that is correct. Which of these molecules stores genetic information? The only one that does that is a nucleic acid, and that is 5. I know this is a nucleic acid because I can see the phosphate, the sugar, and the base. Which of these molecules are used for chemical reactions and structure? I know that proteins do that, and I can see 2 amino acids here. Number 1 is an amino acid. I know that because of the amine group, the carboxyl group, and the R chain. And the same is true with 3, amino, carboxyl, and the R chain. Which of these molecules has a polar region and is found in cellular membranes? This, of course, would be the phospholipid. You can see that here with number 4. We have our fatty acid chains, one saturated, one unsaturated because of that double bond. And you can see the phosphate group right there that gives it its polarity. So we've now gone through all 4 of the macromolecules. You should now be able to identify each of the 4, explain the monomers that make it up. Carbohydrates have monosaccharides, lipids have a glycerol and fatty acid chains, proteins have amino acids, and nucleic acids are made up of nucleotides. And hopefully you feel comfortable in describing the bonds. We're dealing with hydration and dehydration reactions with carbs, lipids, and proteins, and nucleic acids have that unique phosphodiester bond that makes up their backbone. I hope this series of lectures was helpful in understanding the 4 macromolecules, and I will see you all soon.